Sir, title of the topic is comparative phytochemical profiling and in vitro evaluation of some curcuma species using spectroscopic and chromatographic techniques. I am Mrs. Zagde Aditi Vinayak. Now, introduction of the phytochemicals. Basically, phytochemistry is the study of chemicals produced by the plants and the different constituents of the plants, which is mainly used in a different types of disease as well as disorders. And these different kinds of phytochemicals or phytoconstituents used in a various kinds of industries, nutraceuticals as well as dietary supplements industries. Then the phytochemicals are owing the constituent threat microorganism, environmental hazards to public health, and the significance of phytochemistry in the medical and pharmaceutical industry for the quest for the discovery of new drugs has overshadowed for essence of their industries. Basically, sir, my plan is to use different kinds of spectroscopic and chromatographic to evaluate the various types of phytochemicals or various types of species of curcuma. Medicinal plants have been rich in the source of bioactive compounds, that is, the different kinds of bioactive compounds which are present in the different kinds of medicinal plant and which having the significance pharmacological potential. Comparative phytochemical profiling plays a crucial role in understanding the chemistry, diversity, and therapeutic values of a different plant. Advanced different techniques, that is analytical techniques such as the spectroscopic and chromatographic techniques. In the chromatographic techniques, HPLC, HPTLC, TLC method provides a reliable tools for identifying and quantifying the bioactive constituents. That is, what are the different chemical constituents in the present in the plant, and after identifying what is the exact quantity of the particular chemical constituent present in the plant will be identified by the different analytical methods. Then this study focus on the comparative phytochemical analysis of the selected medicinal plant, aiming to assess their chemical composition along with evaluation of their in vitro biological activities. Then the finding will contribute the scientific validation of their medicinal properties as well as potential of pharmaceutical applications. Then, sir, this is the review, brief review of the work already done in this field. They find the different kinds of antioxidant property, cytotoxic property, as well as any other properties, anti-cancer properties. Sir, plant of this study, that is the Curcuma xerodia. First species of the curcuma is a Curcuma xerodia. This Curcuma xerodia commonly it is a white turmeric and it is the most important crude drug belonging to family called as Zinziberaceae. And genus Curcuma, traditionally it has been reported to possess the many biological activity. It is mainly used for the different kinds of therapeutic action due to presence of phytoconstituents. Different kinds of phytoconstituents present in this species, that is the Curcuma xerodia, that is the primary and secondary metabolite. These primary and secondary metabolites mainly responsible for the growth, development, and reproduction of the different kinds of activities. Then main component of the plant are starch, curcumin, essential oil, and Arabic gum. Another, that is the Curcuma cassia. Second chemical constituents of the curcuma, it is the Curcuma cassia rocks. It is a rare perennial herb having the medicinal properties, and it is basically found in the northeast and central India. The rhizome of Curcuma cassia is a bluish black in color and has a pale yellow flower in reddish border. Basically, it is aromatic in nature and it is useful for different kinds of medical treatments, and it having the different kinds of medicinal properties, as well as the Curcuma cassia contains or it mainly constituents different kinds of carbohydrates, proteins, amino acids, steroids, different kinds of glycosides, flavonoids, alkaloids, tannin along with the phenol. Then third species of the curcuma, that is the Curcuma amara, commonly known that it is also called as the mango ginger or haldi in various parts of India. Curcuma amada is a member of ginger family called as Zingiberraceae and is a native to South Asia. It is an herbaceous perennial plant and widely cultivated in a tropical and subtropical region for its medicinal and culinary properties. Basically, the bioactive compounds, especially curcumin and curcuma amida, are well known for their anti-inflammatory activity. That is, these curcuma amida mainly widely used in a different kinds of inflammation or wound inflammation and other kinds of inflammation purpose. It has been used to treat the conditions such as the arthritis, like rheumatoid arthritis, then joint pain and muscle inflammation. Any kind of muscle inflammation, we can use the curcuma amidia. It mainly act as a muscle relaxant also. Then next, objective of this study. The primary aim of this study is to evaluate phytochemical composition and pharmacological activities of Curcuma cassia, Curcuma amida, and Curcuma xerodia for focusing on or by focusing on identifying and quantifying bioactive phytoconstituents such as alkaloids, flavonoids, and phenolics. Then investigating the antioxidant property, anti-inflammatory property, antimicrobial, anti-diabetic, hepatoprotective, along with anti-cancer activity of plant extract. Identifying markers compounds for the standardization and quality control using HPLC and HPTLC techniques. Then proposed methodologies during the tenure of the research work. Proposed methods of the work. First, collection, identification, and authentication. Identification and authentication includes ensure correct identification using the botanical references or expert assistance. Second, extraction of plant material. In that, washing of the collected material thoroughly in running tap water, then rinse with the distilled water, drying and powdering, grinding the dried material into the fine powder. Then defatting the plant material. The powder plant material is subjected to extraction with the petroleum ether via maceration method. Continue the extraction until the material is defatted. Different kinds of fat will be removed. Unnecessarily fats. Four, successive extraction with different solvents. Defatted plant materials is extracted successfully with ethyl acetate, ethanol, water using maceration technique. Then preliminary phytochemical screening, the extract are tested for the presence of following compounds. Basically different kinds of following compounds or different kinds of following phytoconstituents present in a different species of curcuma, that is alkaloids, glycosides, phenols, tannins, flavonoids, carbohydrates, proteins, and sap. Then estimation of various phytoconstituents. In that first, estimation of total alkaloids, then estimation of a total flavonoids by UV spectroscopy method, then estimation of total phenolic contents by modified Folin CO culture method by UV spectroscopy. Then seven, in vitro antioxidant free radical scavenging assay DPPH mode. By using the DPPH model, I am going to assess the free radical scavenging ability. Then NO model applicable for the nitric oxide scavenging activity. Then hydrogen peroxide model used for the scavenging activity. 8. In vitro anti-inflammatory model, albumin denaturation inhibition model to assess the anti-inflammatory activity of different kinds of phytoconstituents present in the curcuma species. Then 9. In vitro antimicrobial activity study. Selection of preparation of microbial culture antibacterial for the antibacterial activity. I am going to use the bacterial strain. Antifungal activity using fungal strain. Then in vitro antidiabetic activity model, yeast glucose uptake inhibition assay, alpha amylase inhibition assay, alpha glucosidase inhibition assay. Then 9.3 in vitro hepatoprotective activity assay of the particular substituents. Alanine aminotransferase inhibition assay, aspartate aminotransferase inhibition assay, then superoxide demonstrates inhibition assay. 9.4 in vitro anti-cancer activity of different phytoconstituents. Conduct assay of anti-cancer potential such as MMT assay or cell viability test. Then identification of marker compound by HPLC. High performance liquid chromatographic technique used for identifying the marker compound in the extract, different extract. Then identification of marker compounds by HPTLC. HPTLC used for the quantitative analysis of the marker compounds. And last, statistically analysis of data by using the different softwares. Then expected outcome of proposed work. Basically, sir, proposed study aims to provide comprehensive understanding of phytochemical composition and pharmacological activities of Curcuma cassia, Curcuma amada, and Curcuma xerodia. One of the key extract outcome is the identification and quantification of bioactive compounds present in these plants such as alkaloids, flavonoids, phenolic compound, glycosides, and tannins. Through detailed phytochemical profiling, this research will provide insights to the specific chemical constituent that contributes to their medicinal properties that is used in a different kinds of disease or disorders. By correlating these compounds with the biological activities, the study will help identify which constituents are responsible for antioxidant property, anti-inflammatory, antimicrobial, anti-diabetic, and hepatoprotective activity along with anti-cancer effect. That is different types of these phytoconstituents or Curcuma cassia, Curcuma amide, and Curcuma xerodia having the different phytoconstituents, and these different phytoconstituents shows their different pharmacological activity. This could lead to discovery of new natural agents that have therapeutic potential. Then these are the different references used for the proposed work.
How you will conduct your experimentation and where you will conduct all those experiments, like
Uh,
chromatography, etc.
uh, Sir, chromatographic technique will conduct in my college also. I am working as an assistant professor in a pharmacy college. There are HPLC UV is available, sir, in our college.
Okay, so you can do all the chromatography and the testing in your institution only.
Yes.
So
Some of the
all the machines in your institutions having are calibrated.
yes, sir.
Okay, who did the calibration for those machines?
The basically, sir, calibration process is I don't know, but after starting the work, all these things I will discuss with the principal, sir, and we'll start the work.
Okay. And what will be the you prefer for the trial of your medicine that you want to be extract?
Uh,
What type of trial you will going to conduct?
Basically, sir, we will check the anti-inflammatory activity and the anti-cancer activity. Some of these parameters or some of this study is available in our Pune Vidyapit. We'll send some samples in a Vidyapit as well as university, sorry, and as well as at my level or in my level, some study is possible in our college only. We'll perform this study in our college, and some sample will be sent in a university.
What about animal testing and others?
Uh, Sir, basically till date, not decided to work on animal study, but my plan is to work on the animal study, different phytochemical constituents used by studying on the animal. It is my plan, sir, but actually now it is not decided, sir, by my side. Basically, it is depends on the sir my guide.
So how many peoples are presently, have you identified the peoples who are working in research in this field? Presently, they are working in the research on the particular, like white ginger, white turmeric, or mango turmeric, what you are saying. People are working on there. What
Uh,
are their identification? What they identified or what they found in their study?
Sir, actually,
कॉल कर रहा हूँ 5 मिनट से. हम्म.
sir, actually, one of my friend is working on such different kinds of phytochemical constituents. He got some new chemical entities or as well as new chemical activities work on the anti-inflammatory activity as well as used in a rheumatoid arthritis, sir.
Okay. Okay. Just all the best. You had prepared well.
Okay, sir.
Okay, but keep in mind that what type of methodology you want to explain, keep everything should explain that everything is mentioned, whatever the process, what are the machinery, they are available with your institution, particularly with your university from where you are conducting your PhD, not
Okay.
in your institution where you are working.
Okay, sir.
Now come to the trials, which type of trials you want to do, you just explain that as well. Key first clinical trial, animal trial, and human trial, how you will bring all those things and what minimum requirement you can fulfill during your PhD that you have
Okay.
to be mentioned.
Okay. Okay, sir.
Okay, so
Okay.
that are the things that you have to be explained them.
Okay,
Okay,
sir.
okay. All the best.
Thank you. Thank you very much, sir.